Hi, and welcome to Chapter 10, Muscular System. So today we're going to explain the organization of our muscular tissue. We're going to describe the function and the structure of skeletal, cardiac, and smooth muscle. We're going to talk about how muscles work with tendons to move the body. We're going to describe how muscles contract and relax. We're going to define the process of muscle metabolism. We're going to explain how the nervous system controls muscle tension. We're going to relate the connections between exercise and muscle performance, and we're going to explain the development and the regeneration of muscular tissue. So there's three types of muscular tissue. There is skeletal, smooth, and cardiac, and all of those are going to arise from the mesodermic embryonic layer. So if you remember, we talked about how there's the endoderm, the mesoderm, and the ectoderm. So this is going to be the center layer. So skeletal muscles develop in a pretty cool way. Okay, so we have these satellite cells. Satellite cells are like stem cells. Okay, and they can remain stem cells. Okay, but some of them are going to differentiate into myoblasts, and then they can also then start to fuse together. These myoblasts start to fuse together, and they eventually become myotubes, right? And so these little black dots are the nucleus. And so you can see that as they fuse together, they keep their nucleus, right? And so that's how the skeletal muscle is going to become multinucleated. You can also see that we retain some of those satellite cells for any future repairs that need to be done. Okay, so skeletal muscles, this is a picture of skeletal muscles. They're going to contract in order to cause movement. They stop movements in the means that they resist gravity, right? So as we're just sort of standing or even walking, they resist that gravity. They prevent any excess movements at our joints. They maintain our skeletal structure and stability, and they help to maintain homeostasis because they generate heat. Okay, so you can see the skeletal muscles, they are striated, meaning that they have these stripes. You can see them kind of moving in this direction. They're multinucleated. There's the nuclei along the edges. Okay. Each of your skeletal muscles is actually its own organ, and they join together to work as one to become an organ system. The organ itself, it actually includes the skeletal muscle fibers, but also the blood vessels, the nerve fibers, and the connective tissues, right? So let's talk about those connective tissues. There's three connective tissue layers. There's the epimysium, the paramysium, and the endomysium, okay? So the epimysium is going to be on the outer edge of the entire muscle fiber, okay? So here's the muscle, that whole skeletal muscle. You have the epimysium on the outside, okay? Then you have all of the paramysiums, okay? They're going to surround each muscle fascicle, okay? And then around each muscle fiber is going to be the endomysium, okay? So here you can see it if you just imagine for a second that you had a bunch of spaghetti noodles and a bunch of straws. And you put as many spaghetti noodles as possible within a straw. Okay, so just picture it or do it if you want. Go get a straw and some spaghetti noodles and give it a try. Okay, and so each of the spaghetti noodles is going to be the skeletal muscle fiber. Okay, which apparently my mouse isn't working. There we go. So there's the skeletal muscle fibers. That would be the spaghetti noodles. And they're shoved into a straw. And the straw is going to be the paramysium. Okay. And now I want you to imagine that we took a bunch of the straws filled with the spaghetti noodles and we wrap it up into plastic wrap. And that plastic wrap is going to be the mycelium. And that's sort of that idea of the muscles. Okay. So here's just a little diagram for you. So here's the muscle. The muscle is coated in the epimysium. Okay. Each fascicle, that's the straw. Oops. Back it up. Okay. So here's your muscle fibers. Okay. Those are your spaghetti noodles. Okay. And there they are. Inside your straw. Your straw is covered over. That is a muscle fascicle. is covered over paramycium. Then we have our muscle fiber cells. Muscle fiber cells are coated in what's called the sarcolemma and the endomycium. Okay. Then we have our myofibrils, filaments, and the sarcoplasmic reticulum. And we're going to get into all that. So the sarcolemma versus the endomycium. So you might see these as interchangeable, okay, but they're not technically. The sarcolemma is the cell membrane. So the muscle cell is covered, remember, with that cell membrane, and that's called the sarcolemma when we talk about our muscle fibers, okay? And that sarcolemma is then surrounded by the endomycium, okay? Each muscle cell that we have is actually densely packed together with many rod-like elements, and those rod-like elements we call myofibrils, okay? This would be an example of a myofibril. The myofibril are going to run parallel to its length. You could imagine this would be like the flour within our idea of our spaghetti noodles. Okay, and it's going to contain our sarcomeres. And sarcomere is the actual contractile element of each muscle fiber or each muscle cell. Okay, and there are striations on each muscle fiber cell. There's a light or an I band, I is in the word light, or there's dark A bands, and A is in the word dark. So the videos have been crashing when I try to play them, so I'm going to skip it for now, and I will link it for you. So this is a sarcomere, okay? And the sarcomere is the functional unit of that muscle fiber or the myofibril. So as the myofibril contracts, the whole muscle cell contracts, okay? So what we do is we have a contraction here at each sarcomere, and that's going to cause the whole muscle cell to contract, which therefore causes the entire muscle to contract. Okay? So if you just imagine for a second that you're all lined up at like Red Rover style, right? So everybody's holding hands in a line. And if each one of you were to pull your hands together, like your arms down, okay? And when you do that, that causes everybody to get a little bit closer, okay? And so it's that idea. So we contract here at the sarcomere. And if we, here's another sarcomere over here, another one over here, okay? And so each person or sarcomere contracts their arms, we move closer together, okay? And so remember, the sarcomere is that functional unit, okay? And so since we have that muscle myofibril contracting, the whole muscle cell contracts, okay? The sarcomere is formed by the lighter I bands and the darker A bands, okay? The lighter I bands are actually formed, they're called a thin filament, okay? And the thin filament is molecules of actin, okay? And then we have the thick filament, okay? The thick filament is going to be our darker A band, and that thick filament is made out of myosin. Okay? And so on the actin, we have what is called tropomyosin, okay? And here we have our myosin, okay? Myosin has what we have on them, it's called an actin binding site. It's like a little head here. And on our actin, we have a binding site for myosin. So if we just back up really quickly, this is our neuromuscular junction. So every place that we have a muscle fiber, it is innervated by a neuron. Okay, so this is a neuron, and it comes together at a neuromuscular junction. You might see it abbreviated as NMJ, neuromuscular junction. Every skeletal fiber in every skeletal muscle is innervated by one of these motor neurons. Okay? And so what happens is we have a stimulus, and that stimulus is going to cause these vesicles that contain neurotransmitters to release their contents into this area. Okay? And this area is called a synapse. And when that happens, the neurotransmitter is going to bind to receptors on the muscle fibers. Okay, and so the excitation signals from the neuron are the only way to functionally activate the fiber to contract. So we have to have this happen in order to get this muscle fiber to contract. The way that we get all of this to happen is through what we call membrane potential. So certain cells, so specifically your neurons, your nervous cells, and your muscular cells, and some endocrine cells have what we call a membrane potential. So the inside of the cell is usually about -60 to -90 millivolts relative to the outside. We call this a resting membrane potential, meaning that it has the potential to do something. Okay? Neurons and muscle cells are going to use that membrane potential to generate our electrical signals. And they do that by controlling ion movement. Okay, so I'm just going to switch to a whiteboard real quick. Okay, so when we have our cell, sorry, it's terrible drawing. The cell, we always say, is okay. And that's because within the cell, we have a lot of potassium. Okay. Now, also within the cell, we have a negatively charged protein. Now, I'm going to make it the P for protein, but most often you're going to see it as an A. Okay. So this is a negatively charged protein in here. And then over here, you have a bunch of sodium. Okay. All right. So what happens is, now this is in here is really negative inside this cell. And over here, it's real positive. Okay. Relative, right? So it's all relative. So what happens is this cell that has this high amount of potassium in here, which by the way is staying like this because we have what's called the sodium potassium pump. Okay. And so what it's doing is it's kind of keeping this at its normal stage. So you guys see that again? No, let me share this back out for you. Okay. So here, right, you can see that the sodium at rest, the potassium, it can move pretty freely across, right? But it's super attracted to that negatively charged protein. And because it wants to be in there, because this is positive and opposites attract, so the positive is attracted to that negatively charged protein, it's like, let me in, let me in. So it just keeps moving over there, right? But I forgot to add the chlorine on mine, the chloride ion. And then there's the sodium out here as well, right? And so these two, they're not really crossing that membrane very easily at all, okay? Also, this negatively charged protein can't move, right? So we have that sodium potassium pump, and that sodium potassium pump is going to pump 3 sodium out and 2 potassium in, right? So 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in. And so it's going to help to maintain this resting membrane potential. How do I get that? So here, right, we have cell and it is okay, right? This is supposed to be a circle. I apologize. And then out here, we have our sodium. Okay. And so what happens is, remember, we have that binding of the receptors on our nervous cells, right? So we had our neuromuscular junction. This is a neuron, right? Inside of this neuron is a bunch of neurotransmitters in there, okay? And those neurotransmitters are going to be released into this area we call the synapse, okay? What happens, it's going to, that neurotransmitter, which often in this case is acetylcholine, it's going to bind to this receptor here, okay? And that is, it's called a ligand gated channel, L-I, it's really hard to write with your finger while holding a mouse. Ligand, L-I-G-A-N-D, okay? Ligand gated channel. So that neurotransmitter right here, it's released out at this neuromuscular junction, okay? And it's going to bind to this ligand gated channel. Okay. When that happens, it is a sodium ligand gated channel. And so the sodium is going to be like, oh my gosh, you opened the door. Let me in. Because remember, in here is super negative. And sodium sodium is positively charged, right? And so the sodium's like, let me in, let me in, let me in. And so tons of sodium is going to move in, okay? And so if we just imagine here, this is our electrical charge, right? Is that we started out really negative, okay? And then we let the sodium in. And if you let in a whole bunch of positive, we're going to move up to a positive number, okay? And so what happens is, as that sodium moves in, we hit this number of negative 55 millivolts, right? So we were hanging between negative 60 and negative 90. On average, it's about negative 70 most of the time, okay? And so we bound to that sodium channel. That lets sodium in, right? Once we hit this number, which negative 55 millivolts, we call that number threshold. When we hit threshold, okay, that is going to open up our voltage gated sodium channels. Okay, so we're opening up even more sodium channels. And so now even more sodium can flood in. So when more sodium can flood in, we continue to move up positive, right? And so sometime around about positive 30 millivolts, okay, those voltage gated channels are going to close off, okay? And we're going to be like, okay, we're good. We don't need to have them moving in anymore. Okay, so there's my cell. Now my cell is got a lot of sodium in here and also a lot of potassium still, right? The potassium didn't go away. I don't know what I did. There we go. The potassium didn't go away. Okay, but now we've closed down those channels. Okay, we're up here. This is my chart, my graph. So there's a bunch of sodium in here, a bunch of potassium. Now it's no longer super negative in here. Now it's super positive, right? And so what happens is when we get up to this peak, my channels are going to sort of flip-flop, right? So I allowed a bunch of sodium to come in, but now my sodium channels are going to close because they were voltage gated, and my voltage has changed significantly, right? I've gone from negative 55 all the way up to positive 30. And because I've done that, I am going to close all those voltage gated sodium channels. And now I'm going to open up voltage gated potassium channels. Okay. And so those voltage gated potassium channels, when they open up, where you want to go? If you're super positive and it's super positive in here, I want to leave, right? I want to get away from the positive. So the potassium is going to leave now, right? And so the potassium is going to keep leaving, and we're going to bring ourselves down. There's not supposed to be a hump there. I apologize. And we're going to come down, but we're going to come a little bit too far. We do what's called hyperpolarization. So this is called depolarization over here. Repolarization, hyperpolarization. Okay, so we go a little bit too far, okay, before we close down my channels, okay? So now I've got a bunch of sodium in here. And now my potassium is out here. My cell is no longer okay. Here's my chart, which is a terrible chart. Okay, so I have to get myself back, right? And the way that I get myself back is the sodium-potassium pump. Sodium-potassium pump. So that sodium-potassium pump is, hold on, getting you back here. Hi, guys. That sodium-potassium pump, it's going to move 3 sodium out and 2 potassium back in, right? So 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in. And it's going to put it back like this. Okay, and so that is an action potential, right? It's an explosion of electrical activity. Okay, so here I am resting at about negative 70 millivolts. Okay, so we get this depolarizing current, or we get a binding. Okay, so we get those sodium channels open. So remember, we had to bind to those ligand-gated sodium channels. And when I bind to those ligand-gated sodium channels, my sodium is going to move into the cell, right? Because it's attracted to that negatively charged protein. And sometime around negative 55 millivolts, that's this part right here, negative 55 millivolts is threshold. And so once I hit that threshold, now I'm going to open up my voltage-gated sodium channels, okay? And I'm going to keep letting the sodium in. This is like the idea of like a concert, right? So the first ticket holders, the first sodium ticket holders, they can get in right now, okay? But at a certain time, that's when the rest of the ticket holders can come in, okay? So we open up the gates and those sodium, and the rest of those sodium channels open, okay, until we go a little bit too far. So this is depolarizing, okay? Now the sodium channels close, because remember, those are voltage gated, and we've changed voltage. Okay? Now our voltage gated potassium channels are going to open, okay? And so the potassium is going to flood out of the cell. So we start moving back towards negative. And now my voltage gated potassium channels are going to close. And now our sodium potassium pump is going to put us all back to normal. Okay. This idea of threshold is called an all or none principle. If we don't reach threshold, we can't make this happen. But once we reach threshold, we can't make it stop. So all of that is like the first step of what we call the excitation contraction coupling. Okay, that neurotransmitter, it released, so this neuron released it to the neurotransmitter into the synaptic cleft. We call it a synapse or synaptic cleft. Okay, and that neurotransmitter here that we're using is acetylcholine. The acetylcholine bound to the receptors on the sarcolemma. Remember, the sarcolemma is the cell membrane of a muscle cell. Okay, and that whole idea, right, is that when we bind that acetylcholine, that's sort of the stimulus to really get this started. Okay, and that's going to cause the opening of the sodium channels right here. Okay, so we bound to that receptor. The acetylcholine has bound to the receptor. We're going to open up the sodium channels. The sodium is going to move in. Okay, and when that sodium starts to move in, we're going to hit threshold. We're going to hit those voltage gated channels so even more sodium can flow in. Okay, that voltage gated sodium channels are going to close. The voltage gated potassium channels open. Okay. We now are heading back negative. We're repolarizing. Okay. We've gone a little bit too far. We've hyperpolarized. Those potassium channels are going to close. Now the sodium potassium pump is going to put us back to normal so that we can be depolarized again. Okay, and so that all of that is called like an action potential. So that action potential is generated and it's propagated. Propagated means that it moves along the sarcolemma. Okay, that sarcolemma again is the cell membrane of the muscle cell. That's going to travel down what we call a T-tubule. And as it travels down the T-tubule, we're going to release calcium ions because calcium ions are surrounding the T-tubules. They're hiding in this area called the sarcoplasmic reticulum. Sarcoplasmic reticulum, very similar to the endoplasmic reticulum, right? So sarcoplasmic reticulum is holding all of that calcium in there. The calcium ions are going to bind to troponin. Now remember that troponin is on the actin or our light I bands, okay? And when that happens, the troponin is going to change shape. And when it changes shape, it removes the blocking action of the tropomyosin and it lets the actin become active, right? It has these actin active sites. They are now exposed. So that means, okay, that the myosin cross bridges can start to form. They alternately attach to the actin and they detach, and they pull the actin filaments towards the center of the sarcomere. Okay, so they start to attach here and they start to pull, and this continues to sort of rotate around. We call it a power stroke. Okay, and we can start to contract. Okay, we get this release of energy by ATP here, and that is hydrolysis powers the cycling process. That ATP is powering it. Okay, then we have to get some removal of the calcium by active transport into the sarcoplasmic reticulum. When we start to remove that calcium, the tropomyosin blockage becomes restored. Okay, so then we block that actin active site again. The contraction ends and the muscle fiber can release. Okay, so that happens at that motor end plate, okay, at that neuromuscular junction. So here is that whole idea, right? So every one of these muscle fibers is innervated. They have that neuromuscular junction. Okay. This is how that propagation happens of sending that action potential. This is happening within a neuron, okay? But the same idea happens in that sarcolemma, right? Is that this area is depolarized and we start to move that action potential down. So as this part becomes depolarized, this is repolarizing, okay? And so it continues to travel down, okay? That plasma membrane. So the contraction of the muscle fiber actually starts when the action potential reaches this neuromuscular junction. So right here. And it stops when the signal stops, when we stop telling our muscles to move, or when the muscle ATP actually runs out and the muscle becomes fatigued. This is how we relax it back. We've already talked about that a little bit. This is called the sliding filament model of contraction. Okay, so remember that the muscle fiber shortens along the sarcomere. The sarcomere is that functional band. Okay, and as it shortens, the myosin heads pull on the actin filaments so that we can continue to shorten and contract. Okay, the A bands are the dark ones, the light bands are the I ones. Okay, the light bands we also called actin, and the A bands are dark, we call myosin. Okay, so when the sarcomere contracts, the Z line right here is going to move closer together and the I band becomes smaller. The A band here is going to stay the same width. At full contraction, these thin and thick filaments are going to totally overlap one another. This is just another visual to sort of show you what happens. You can review it in the slides that I posted for you. Okay. So muscle metabolism happens in three different ways. So first we have direct phosphorylation. Direct phosphorylation requires no oxygen. Okay, and we can do this for about 15 seconds. And this is when we get the reaction of creatinine phosphate, or CP, and ADP. Okay. We also have an anaerobic pathway. The anaerobic pathway can happen for about 30 to 40 seconds or even slightly more than that. And we do this through glycolysis and lactic acid formation. Okay, so the glucose, which we use from the glycogen breakdown, or we get it from our blood. Okay, the glycolysis happens within the cytosol. We get out some ATP. We then get pyruvic acid, which can then be converted over to lactic acid, and that's going to be released into the blood. Okay. And so this is our anaerobic pathway, meaning that we're not using oxygen. This is the best choice when we talk about an energy production standpoint. This is the aerobic pathway. This is aerobic cellular respiration. Okay. And when we do aerobic cellular respiration, we're going to take in one glucose molecule, and out of that one glucose molecule, we're going to get about 32 ATP. Okay. And so here, one glucose molecule, 2 ATP. Right? And so we're really looking to get that energy from ATP. Okay, and so we can do this for hours, right? So when we have glucose, we are going to convert it into pyruvic acid. We're going to go into the aerobic respiration within the mitochondria and do all the different steps of cellular respiration, like the cycle. And out of that, we're going to get out carbon dioxide and water, but also ATP, lots of it. So muscle strength is directly related to the amount of myofibrils and the sarcomeres within each fiber. The number of skeletal fibers themselves is genetically determined, and that doesn't change, right? So we're actually just wanting to change the amount of myofibrils and the sarcomeres within each fiber. So we can do what's called hypertrophy. This means that we can get very large. So hormones, stress, and steroids can increase the production. That would be hypertrophy. And then when we have decreased use, or if you have disease that reduce production, we can get atrophy. There's different types of muscle contractions. So there's isotonic contractions. So in isotonic contractions, we're going to contract concentrically. This is when you would think of like lifting up a weight, right? That's a concentric contraction. We shorten the muscle in order to move a load. Okay. We also have eccentric contractions, and this is when we lengthen the muscle. So there's tension reduced, right? So when we go all the way down. Okay. And then we can isometrically contract. And isometrically contract means that the muscle is going to produce tension, but we don't change the skeletal joint angle. So you're just holding it in place, no movement. So this is a myogram of a muscle twitch. It's a graph of showing what a muscle is doing. So there's the latent period, and the latent period means that the action potential is being propagated along the sarcolemma, but the tension hasn't really changed yet. Okay. Then we have the contraction period, and the contraction period means that those calcium ions in the sarcoplasm are going to bind to the troponin. The tropomyosin moves from the actin binding sites, cross stitches form, and then the sarcomeres are going to shorten. Okay, that's the actual contraction. And then the relaxation phase is when the tension decreases because those calcium ions are getting pumped out of the sarcoplasm, and the cross bridge cycling is going to stop. We can get different wave summation and tetanus. Okay, so first this is the tension, and then this is where the stimuli happens, the little arrows. Okay, so a twitch is just a single stimulus is delivered, the muscle contracts and relaxes. This would be something that it's almost impossible to just make your muscles twitch, but often we see this in people's eyes when they get nervous or stressed. Their eye will start to twitch, or their eyelid. Okay. We also can get wave summation and unfused or incomplete tetanus. Okay. And so for wave summation, the stimuli are delivered more frequently, and so you can see that we never really come back down to a relax. Okay. And so the contraction force continues to increase. Then we have our unfused or incomplete tetanus, and this is where we have a more complete twitch fusion because the stimuli are they're delivered much more rapidly. And then we have a fused or complete tetanus, right? And so this is a smooth, continuous contraction without any evidence of a relaxation. There's different types of muscle fibers, and they do different types of respiration. Okay, and so for our long distance type 1, these are called slow twitch fibers. They have a low fatigue rate. So long distance would be like we're going on a marathon. Okay, and those are also called type 1. So type 1 are called slow oxidative. So they are using oxygen. So they're using aerobic respiration. They have fast contractions. Type 2A would mean that we are doing like I don't know, like a 400-meter race. Okay, and this is fast oxidative. They have fast contractions. They use aerobic respiration, meaning that they use oxygen, but they can actually shift to anaerobic respiration. So because of that, they can tire more easily. And then we have super short sprints. Okay, these are fast glycolytic. They have fast contractions, and they primarily are going to use anaerobic respiration. So they're not using oxygen, and because they're not using oxygen, they tire very quickly. This is cardiac muscle tissue. Cardiac muscle tissue is only found within the heart. It is striated, meaning that we're still going to see those stripes because it's organized into the sarcomeres, and it has the bands just like our skeletal muscle does. Cardiac muscle fibers, though, are shorter than the skeletal muscle fibers, and they only have one single nucleus. Cardiac muscle tissue does not fatigue. but it does have what are called intercalated discs. These intercalated discs are pretty cool. Okay? There's the desmosome with the intercalated discs, and that is going to allow for connection between them. And then we have gap junctions in here. Gap junctions allow for electric coupling. The desmosomes are going to keep those cells connected together. Within the heart, we have pacemaker cells. Pacemaker cells are autorhythmic, okay? And they are self-excitable because they are able to depolarize to threshold and fire action potentials on their own, okay? And the reason being is because they hit threshold because they have leaky channels, right? So if our cell is okay most of the time, and we have to have those sodium channel, we have to have a binding for the sodium channels to open, these have leaky sodium channels. So the sodium's kind of leaking in, and then we hit threshold, that's gonna cause the huge jump of those sodium channels, right? Those, the voltage gated sodium channels are gonna open. We can actually transfer the depolarization to our other cardiac cells. So they move from the SA node, which is the sinoatrial node. That's the pacemaker cell. It sets the sinus rhythm of the heart. Then we can have it go to the atrioventricular node. The atrioventricular node is right here. These are the atria. These are the ventricles. So it's right here at the atria and ventricle connection. Okay. Then they go to what's called the bundle branches, and then out from there to the Purkinje fibers. And that allows it to depolarize through so that we can get the depolarization to travel and then cause the heart to contract. The heart is pretty cool because it has what's called sustained depolarization. So remember that in our skeletal muscle cells, we would go up and then we would come right back down, right? But in our cardiac muscle cells, we have a sustained depolarization, meaning that we plateau right here. And the reason that we plateau is because we open up these voltage gated calcium channels and we close some of the potassium channels. We do this so that we're not able to immediately beat the heart again. Okay. And so we have what's called a refractory period. And that means that the heart cannot beat again at this point. That's going to allow the ventricles to refill with the blood before we pump. And then we have our smooth muscle. And smooth muscle is present in the walls of our hollow organs. Okay, so like our urinary bladder, uterus, stomach, intestines, the arteries, veins, walls, the tracts of the respiratory, urinary, and reproductive system, and even in our eyes. They have spindle-shaped fibers and a single nucleus, and they produce their own endomysium. There are no striations, but they still contain actin and myosin. They do not, however, have troponin. Instead, they have what's called calmodulin. Okay. And they're also organized in a little bit of a different way in that we have what's called dense bodies and Z discs. Okay. And so we don't have the C lines, we have Z discs instead. And that allows them to contract, if you like, to the inside, right? So they bunch together and contract like this and sort of interlock and interlace together. Okay. And there's no neuromuscular junctions here in our smooth muscle. Instead, we have what are called varicosities. Varicosities are along the axons at the autonomic nervous system, and they form loose motor units sort of surrounding that. Okay. And so within each of these varicosities, we have these vesicles with neurotransmitters in them. Those neurotransmitters can be released and then bind. This is a comparison of our cardiac, smooth, and skeletal muscle. It's a triple Venn diagram. So you'll see here, this is just skeletal, this is just cardiac, this is just smooth. This is going to be skeletal and smooth together, right? So similarities. This is smooth and cardiac similarities. This is cardiac and skeletal similarities, and then this is all three. So I encourage you to really take a look at this and try to recreate it in your own words. If you have any questions, feel free to reach out. I am available through email, set up a virtual office hours if you would like. Thank you. Bye, guys.